And I would like to introduce Dr. Emma Hapke, who will be the chair for this first session.
Thank you so much. It's a pleasure to be with you all today. Thank you to everybody who's tuning in today. So our first panel, our very first speaker is actually Dr. Sidney Kennedy. So since you just heard his bio and his introduction, I'm not going to repeat that, and I'm going to give him the floor. Go ahead, Dr. Kennedy.
As I've told you, we're going to deal with these three sessions. And I think the point here is really to highlight that we had a very interesting post-war 2 decades with Albert Hofmann, as many people will recognize, playing a pivotal role both in the discovery of LSD and its psychoactive properties, to later on, 15 years later, discussing psilocybin and identifying psilocin as an active ingredient. And there was tremendous buy-in from the psychiatric community. Joel Elkes, a psychiatrist, published on LSD after his own experiences in the early 1950s. A major conference at Princeton, a multidisciplinary event, highlighted the potential benefits of LSD in psychotherapy. Sidney Cohen published a positive meta-analysis on the safety of LSD in 1960. So this was essentially the rise of the psychedelics, but of course this was short-lived. And by 1962, Timothy Leary had conducted his famous experiments at Harvard, randomizing people to psychedelic experience or an active placebo, and subsequently was dismissed from Harvard. Jonathan Cole, a prominent psychiatrist in the 1960s, began to take, as it said, a sober look at psychedelics. And it's worth pointing out that Sandoz Pharmaceuticals actually had been manufacturing and selling licensed products, both in terms of LSD and psilocybin, which they then withdrew in 1965. And of course, by 1970, we had Richard Nixon essentially ending the use even for medical experimentation of psychedelics, particularly LSD, psilocybin. And if we think about the actual compounds, people ask, what exactly is a psychedelic? And here's a definition from Greenspoon, highlighting that a psychedelic should not cause physical addiction, craving, or delirium, but may produce thought, mood, and perceptual changes. Experiences similar to dreams, religious experiences, or acute psychosis. And of course, the question of spirituality has been a main theme throughout the use of various psychedelics. Some individuals have considered the serotonergically acting agents, the tryptamine derivatives, as being the core psychedelics. Others would take a broader perspective. And as I say, tryptamine derivatives would include LSD, psilocybin, and DMT, the active ingredient in ayahuasca. And the phenyl ethyl amine compounds would include mescaline and MDMA. And I've highlighted psilocybin and MDMA as probably the two compounds that have received the most active therapeutic exploration in the past decade. So going very quickly, LSD was not only discovered by Hofmann, but he wrote very eloquently about his experience, and he highlighted issues such as the fantastic pictures, the extraordinary shapes with intense kaleidoscopic play of colors, and the drug was marketed as Delicid in 1949. psilocybin was actually written about in the Florentine Codex in the 16th century. The secret mushrooms of Mesoamerica being considered God's flesh. And of course, one of the first photo essays in Life magazine in 1957, the first recognized publication in a lay magazine, was on seeking the magic mushroom. And again, psilocybin was marketed as indocybin. Moving on, after the United Nations and the US had essentially shut the door on any research, the only survivor was MDMA, which had initially been developed by Leo Zeff. And it continued to be used until 1985, when it was also placed on Schedule 1. But you can see almost immediately that the Multidisciplinary Association of Psychedelic Studies, better known as MAPS, and founded by Rick Doblin, was launched to promote clinical research on MDMA. I won't go into it, but some have considered it, quote, the perfect drug for trauma-related therapy. Ibogaine has received considerably less investigation. It was looked at in terms of potential role in addiction management, but a number of toxicities, unsteadiness, cardiac abnormalities led to it being classified as Schedule 1, and it has not received the same application. In 2000, the New Times highlighted this is coming. Psychiatry may never be the same. Psilocybin and MDMA are poised to be the hottest new therapeutics since Prozac. Universities want in, so does Wall Street. Some worry a push to loosen access could bring unintended consequences. So how did we move forward? And again, very briefly, and I'm not doing justice to many other contributors, but the work that was done at Johns Hopkins by Roland Griffith and many of his colleagues was really continued through what some might call the dark ages of psychedelic research, highlighting individuals who did not necessarily have depression, but also in particular people who were looking at end-of-life care. And there were a number of publications along these lines, and many have actually grown in the last 5 years. But in the UK, Robin Carhart-Harris, David Nutt, and other colleagues essentially applied the technologies of today, including functional neuroimaging, to understand the mechanism of action in the brain of psychedelics, and applied some of the clinical trial designs to evaluate outcomes. I would also recognize Michael Pollan's book, How to Change Your Mind, The New Science of Psychedelics, as making a significant contribution to essentially taking the psychedelic interest to a larger global audience. And the work that's been done in the last decade in terms of how you can actually receive treatment with psychedelics, the major way is to be in clinical trials. And the two major organizations would be MAPS that I've mentioned, and Compass, which is Navigating Mental Health Pathways. And we are fortunate today to have Professor Guy Goodwin, formerly at Oxford University and now the Chief Medical Officer of Compass, to also provide some detail. So I'm going to leave with a few themes that I hope we can give some thought to today. I think the medical community is a diverse and heterogeneous community, and there is everything from healthy skepticism to what some might consider blind adoption. I think there are questions of how we design studies, how we manage blinding, in a number of the trials, which is not peculiar to this area, but certainly is an important issue in general. We haven't traditionally seen licensing of substances or new drug treatments based on a combination with psychological therapies. So there is a bit of a need for a reset on how trials are carried out and reviewed. Many of us have funding structures that are based on time-limited activities. As it currently stands, there are significant funding questions around extended sessions that you'll hear later in some of the presentations. Currently, it would be a criminal act to be actively providing psychedelics in a medical clinic. How do we manage the decriminalization? You'll hear some discussion about the Section 56. We also have to remember the indigenous heritage of many of these compounds, and how do we actually reflect on that, and how do we recognize some of the religious freedom challenges. And finally, I think one of the issues that has been of some discussion is if we're using psychedelics and they're linked into end-of-life care, are we in fact pathologizing dying? And these are discussion points that I hope we'll consider later. So thank you very much for your attention at this stage.
Thank you so much, Dr. Kennedy, for getting us started today and giving us more information on the history of this field. Next up, we have Dr. Gregor Hasler. He is a Swiss psychiatrist with substantial experience in psychedelic-assisted psychotherapy. He was trained in psychopharmacology and neuroimaging at the NIMH in the States. He is currently a professor of psychiatry at the University of Freiburg in Switzerland and president of the Swiss Society for Pharmacovigilance in Psychiatry. His research is supported by the Swiss National Science Foundation. Go ahead, Dr. Hasler.
Yeah, thank you very much for this nice introduction. So thank you very much for this nice introduction. Do you hear me? Just to check.
Yep.
Yes, we do.
Okay. Next step is the sharing of my slides. Do you see my slides?
Yes, we do.
Okay, we're all set. Now it's a pleasure for me to talk about psychedelics and neuroplasticity. Just a moment. First of all, what is neuroplasticity? As you can see here, I mean, in general, that's like the adaptation of the brain to the environment. It involves learning. And there are many, many mechanisms involved in neuroplasticity. You see on the left side, there are molecular mechanisms like gene transcription, epigenetic, things we are beginning to investigate. And there are mechanisms at the cellular level, for example, neurogenesis, a rare event in the human brain, then dendritogenesis, very important, related to psychedelics, and then what we call functional plasticity, that's synaptogenesis, that's LTP, LTD, so it means neurons that fire together, wire together, very basic mechanisms of learning. You see why we are interested in psychiatric research in neuroplasticity. For example, animals after prolonged stress show a decreased rate of neuroplasticity or ability for neuroplasticity. You see, for example, on the right side, you see the branch of a dendrite, you see the spines, and there is a reduction in these spines. These spines allows the cell to connect to other cells. So there's less connectivity, less learning. Maybe that's a protective measure of the brain in a very adverse environment. But we know that on the long run, that causes problems, inflexibility in behavior, inability to learn from rewards, for example, and finally leading to depression. On the left side, you see another concept. When you look at the whole dendrite, you see, we call that a reduction in the complexity of the dendrite under prolonged stress. as you understand me right, a little bit of stress rather enhances neuroplasticity, but prolonged stress leads to reduction in neuroplasticity. And here what's important, I mean, that's studies in animals. Most neuroplasticity research is done in animals. But if you look at the brain tissues in postmortem studies from depressed individuals compared to healthy controls, you see that you can measure, for example, spine density with electronic microscope or electron microscopic images, and you see you have just a 50% reduction in these spine synapses. So that clearly argues for also neuroplastic deficits in patients with major depressive disorder. So here you see the new model, like the neuroplasticity model of pharmacotherapy in depression. You see the healthy brain, and due to genetic and environmental factors, you have cortical atrophy leading to depression with retraction of dendritic branches and loss of dendritic spines. And these structural changes are reversed by psychoplastogens. That's like a new fancy word for psychedelics. But what is then another argument? I mean, we had, or early on for the neuroplastic model of antidepressant response, you can see here. So if there would be just a simple deficit in serotonin, and for example, SSRIs would reverse this deficit, you would expect to have a very fast response, like with benzodiazepine. So you give them, and after an hour or 40 minutes, you see an anxiolytic response. But that's not the case with antidepressants. It takes days or weeks to see the response. The other fact is that, for example, ketamine, one of the psychedelic-like substances, if you give this an infusion to patient, you have a response after 2 or 3 hours. And these responses lasting over 7 to 10 days. Also, the drug is removed from the body after like a day. So to explain this, we cannot use a simple neurotransmitter model, but a more complex model, for example, the neuroplasticity model. Another thing is psilocybin. We heard about this there in some studies. Like here, you see they gave, mainly we would say, a single dose. They gave 2 doses, a test dose of 10 milligram, and after some days, 7 days, 25 milligrams. And then they saw improvement in depression in some individuals over 3 months. You see there are also a lot of non-responders, and certainly that's not a controlled study, but that's certainly a very promising finding. So, but the real fundament of this idea that psychedelics are psychoplastogens comes really from animal studies. One is in vitro studies, as you see here, where you have a cell cultures from rat embryogenic neuronal cells, and you add the substance we heard of from Dr. Kennedy, for example, DMT, a component of the ayahuasca brew, or LSD. Usually LSD has the most potent effect, and you see the number of branches, the number of primary dendrites, and also the total dendritic length increases. You see a schematic illustration of these effects. And the study, they also looked at in vivo. They looked in animals, various species, Drosophila, rats, and they also found this effect in the living animals. So that seems to be a very conserved mechanism found across evolution. Here you also see that ayahuasca, just a little remark, there you have ayahuasca brew with DMT, but you have also the ayahuasca plant, and this plant doesn't contain any DMT, but a lot of alkaloids like harmol, harmine, harmaline. And also these substances increase, for example, neurogenesis in cells from hippocampus. In many studies, there are hippocampal cells because they're neurogenesis in the rodent hippocampus. And we also know in humans that the hippocampus is very sensitive to stress and shrinks in depression. So that also shows that maybe there's more complexity to the ayahuasca brew than just DMT. Here you see now a study in an animal, in rat, I think, and they are in mice. And here they gave a 5-MeO-DMT, that's a particularly potent form of DMT, causing severe ego dissolution, very short-lived, about 2 minutes to 2 hours. Therefore, it's not used in psychotherapy, but it's an interesting substance because it's in this study stimulated cell proliferation and dendritic complexity in adult mice ventral dentate gyrus. So that's a part of the hippocampus. So we see these red spots in the lower figure. These are the new cells. You see that's an increase about more than 100%. And you see these new cells on the left side have more dendritic complexity than the cells produced in the untreated animal. The next step is to see really in the animals, does this enhancement of neuroplasticity really lead to behavior change? For example, an increase in sociability. You measure the various measures, how much they interact, how much they prefer to be with other animals. And you see in this study, a repeated application of LSD, in fact, lead to an increase in sociability. What's interesting here, that's on the right side, you see this mTOR, mTOR. That's a cellular molecular marker intracellularly for neurogenesis. For example, also ketamine increases or works on this pathway. And you see there is a marked increase in this marker, and in this study, this was correlated with the increase in sociability. So indeed, it seems that this neuroplasticity leads finally to, for example, more prosocial behavior when given in a positive social environment. So this is now one of the most complex studies published in Nature, but a very interesting studies where they looked at critical period. So critical period is an important term in neuroplasticity research. It refers to a time during postnatal life when brain learns rapidly from environment. We know that learning accents in humans, so it's easy as a child to learn an accent of a language, and later in life it's very difficult. We also think that it applies to social behavior. If you haven't learned any stable social relationship, any rewarding social interactions, then it's more difficult to learn them later in life. And we know here that this critical period for social reward learning depends on oxytocin. With age, oxytocin sensitive neuroplasticity decreases. So there's the same amount of oxytocin, but the cells responding to oxytocin gets less sensitive, and this is paralleled by the closing of this critical period. And we also know that MDMA stimulates oxytocin neurons. You see it on the right side. And so what they did in a complex series of experiments, they showed that MDMA induced synaptic plasticity in the reward system via stimulation of oxytocin neurons, not via the oxytocin receptor, but on other ways. And they showed that SSRIs and cocaine did not lead to this effect. Even SSRIs added to MDMA blocked this effect. And they also showed that this prosocial effect through MDMA only happens if there is a positive social environment. So the effect was blocked by, for example, by social isolation. So they conclude that MDMA has the potential to reopen the critical period of social reward learning when given in the right setting, leading to prosocial behavior in mice. They call, therefore, MDMA a metaplastic drug. They say it's not really that it directly increases neuroplasticity, it rather creates a context for neuroplasticity to occur. That certainly shows how important the setting is. That's in general a remark that neuroplasticity is not per se a positive thing. It's only a positive thing when you're in a positive environment. In a very adverse environment, it may be even a vulnerability factor. And finally, as the last animal study I'm presenting to you is that these psychedelics are not just interesting for psychiatry research, but also for neurology. For example, here you see they induced a infarct in the brain through ischemia. And then they treated patients with DMT. And you see on the left side, these led to a reduction in the lesion volume. On the right upper side, you see this lead to an increase in BDNF in the serum. I just tell you that's important because that's a marker we use in humans, and we're not so sure if that's related to neuroplasticity in the brain, but in this study it was. But most importantly, on the right side, lower figure, you see that in fact, the forelimb motor function improved considerably giving DMT. You must see in neurorehabilitation, there is almost no evidence-based treatment. So all what they do is not proven to be effective. There's no really substance. Sometimes they use SSRIs, but this would be really a revolution to have compounds that increase neuroplasticity after stroke. So let's go to humans. There is a group around Professor Matthias Lichty in Basel and the city of LSD. They're really looking at LSD, and you see here they gave various doses to healthy subjects, and they found that in fact BDNF increases in a dose response type relationship. So always if you see such a relationship, that's a good sign. That means there may be really a causal relationship between LSD and this increase in BDNF. So we don't know exactly what it means, but in certain, like the animal studies showed you, it seems to really linked to also BDNF in the brain, an important growth hormone in the brain related to neuroplasticity. Another way to look at neuroplasticity in humans is to look at sleep EEG. There are slow waves, and they're related to neuroplasticity during night. Very important because, for example, some of the memory consolidation, some of the housekeeping in the memory is done during night. And you see that ayahuasca had the potential to increase this slow wave sleep. And you see a kind of a clever comparison. Amphetamine that is used for learning disability as neuroenhancement doesn't increase this kind of neuroplastic marker, let's say. So finally, I think what I showed you is there is, in fact, a potential that psychedelics could have neuroplastic properties. And I also showed you that's like a vulnerability to increase neuroplasticity. Therefore, it's so important that the patient is not in a highly stressful environment in his life, but also the environment for the treatment must be well adjusted to the situation. You see, there's usually we take a setting like a living room style room, and you see two sitters, very empathic persons who help the patient with this extraordinary experience. So there's a lot of guidelines also for MAPS how to do this kind of treatment. So thank you very much for your attention.
Thank you so much, Dr. Hasler. That was really fascinating look at neuroplasticity, and really, I think, brings up a lot of important questions. We think a lot about the set and setting in the therapeutic modality with psychedelics, but I think we're also need to be thinking about what about the communities and the social context in which our patients are living and how that might be influencing them after their brains are in these neuroplastic states. So lots of interesting things to talk about when we get to our discussion. Next up, we have Dr. Kim Kuypers, who is affiliated as an associate professor with Maastricht University in the Faculty of Psychology and Neuroscience, where she obtained her PhD in 2003. Her main goal is to understand the neurobiology underlying flexible cognition, empathy, and well-being. To accomplish this, she uses a psychopharmacological model to induce subacute and longer-lasting effects on the behavior mentioned above with psychedelics to investigate the underlying biology. We're thrilled to have you with us. Go ahead, Dr. Kuypers.
Thank you so much, Emma, for the introduction and also the invitation. I will try to share my screen always this in the, oh, wait. Do it like this. Yes, we're here. So thank you very much to give me the opportunity to speak today. I was invited to talk about this hype that is happening next to the psychedelic renaissance. We also have increased interest in microdosing, and I will tell you about that, what microdosing actually is and what we know about it. So I'm very happy that Sid and Gregor already introduced topics that I'm also going to touch upon. And here you see actually a blotter paper of sprinkled with LSD, on which Albert Hofmann is depicted. Albert Hofmann was the father of LSD. He synthesized LSD for the first time. And here you see him on this blotter riding his bike in the Swiss Alps when he was under the influence of a dose of LSD. So, but I'm not going to talk about this large dose. I'm going to talk about a small dose of LSD or other classical psychedelics. And to understand what this is, so microdosing is taking a small amount that does not hamper you in your daily activity. And it's with the intention to actually enhance some kind of effects like enhance productivity, creativity, or mood. So the definition states that it's taking 1/10 of a regular dose, but then you have to question what is a regular dose of LSD. So you can say that between 100 and 200 micrograms of LSD is a regular dose. So 1/10 is then between 10 and 20 micrograms. People take it repeatedly, so not only once. And like I said, it's to improve certain aspects of life, and it does not have these hallucinogenic effects that a full dose has. Here you see how some people take it. So they take it one day and then leave 2 days in between, and then take it on the 4th day again. This is known as the Fadiman schedule. James Fadiman is very important in this context because he introduced this schedule, and he is known for his research on microdosing. But we know from our surveys that people do not only use this schedule, but they come up with their own, taking it with 1 day in between, or every day, or once a week. It is different, and people try to find out for themselves what works for them. So I thought of actually adding this to give you an idea of how people do this. So I've shown you the blotter on the first on the previous slide, and what people then do is they know how much LSD they normally buy, and then they can put it either in alcohol or water, and then take the amount out that they want to take. This is one way they can do it, or otherwise they can just take the blotter and cut some pieces of which they think will be the dose. And we've also shown that asking people, that sometimes adverse effects are that people take too large dose and they experience these hallucinogenic effects. So here is a quote by one person who microdosed, and the person stated that he or she became more productive, more happy, and it induced creativity. And this is important to remember because I will come back to this later in my presentation. I said in the beginning, I was going to talk about a hype, but it is not that new, microdosing, because Albert Hofmann, if you look back in history, he already mentioned the use of smaller doses, and he also alluded to it being potential therapeutic. And if you look back in history, I always love to read old literature. I have to admit it's more difficult to read because they use different methodologies and a different way of writing, but still it is interesting. You see that I think one of the first publications on microdosing was written or published in 1958. And here it is shown. So on the x-axis, you see the doses of LSD. So I told you that between 10 and 20 microgram is microdose. So these can also be considered as microdoses, but they are very small. So the 4 and the 7, the 12, and you see that they induce effects on mood, but also on biological measures like the galvanic skin response, it increases heart rate. So it does something to a person. We asked the question, who uses it and why do they use it? So I already told you why some people use it, but we want to hear it from a larger sample. So we did some survey research, and this is what came out of this research. So people say to use it to increase energy, enhance creativity, as a study medication, cognitive enhancer, to increase concentration. But what is most interesting, I think, to me, is that they also took it for to deal with their psychological and their physical symptoms. So we zoomed in on that, and we asked people, if you compare your microdosing with conventional treatments you ever received, how efficacious do you think it is? But also, if you compare microdosing with a full dose of psychedelics, how efficacious is it in treating your symptoms? And this is what we showed. So when we compared to conventional medication, people said that it worked better, symptoms disappeared, quality of life improved for neurodevelopmental disorders like ADHD. You also see here that it was working for anxiety disorders, for substance-related and addictive disorders. So this already shows the broad range, therapeutic range, for which it could be used. People use it, find it efficacious. But I will come back to that. Is it really true? So when they compare this self-medication with microdoses with full hallucinogenic doses, we see that it is a different picture. If we compare it with this picture, self-medication with microdosing compared to conventional, you see that a lot of indications apply, that they think that it's good for a lot of indications. And if they compare it to full hallucinogenic doses, there are less indications for which they think it works, but still there are some indications. Here you see depressive disorders, still anxiety disorders. So then, as a scientist, I'm really interested whether it really works or it are purely anecdotes or placebo effects. So, but what do we know until now? We know that the anecdotal evidence is much bigger than the scientific evidence, and we've published some papers on this. But we know that there is scientific evidence, and we also contributed to that. I will come to that in some later slides. So how do we do that, collect objective evidence? So we do placebo-controlled studies, which is seen as the gold standard in pharmacological research. So we give people a small dose here, psilocybin is depicted, small dose of psilocybin or LSD. And on another occasion, they get a placebo, and then we assess the effects on well-being or cognitive performance, or even biological markers of neuroplasticity, which Gregor was talking about. And if we see that the effects after a microdose are greater than compared to placebo, we know that it is a causal relationship, that the drug caused the effect. So if we look at recent work, I can put it on 3 slides, what has happened to date. So there's one group in Chicago, and they did microdosing research with LSD. They gave it to healthy volunteers, young, just regular age, I think around 20 to 30. And they showed that microdosing, single dose, has an acute effect on mood, but also on the activity of the amygdala, so the center in the brain that is involved in emotional responses. And they also showed that it does something to event-related potentials, in the sense that they actually have the same effect as a full hallucinogenic dose, but then not so pronounced effect. There is another group that did really microdosing research, in the sense that they gave it repeatedly to people, healthy volunteers. It were elderly people, and they showed effects on time perception, which changed, but that was actually the only effect on behavior they showed. But they also showed that it was tolerable to these people and safe, which is very important if you want to use it as a therapeutic agent. And then lastly, our work, what we did, we gave microdoses of LSD to single doses to healthy volunteers. And what we showed was that it improved mood, also some parts of cognition, it enhanced attention. We showed that it decreased the pain perception of healthy volunteers, which could allude to a benefit in pain patients, which is something we are going to investigate in the future. And Gregor was talking about neuroplasticity. We also showed this for the low doses, for the very small doses of LSD, that it increases neuroplasticity, which can have great potential in the future in the treatment. So I come to my conclusions. What is the main conclusion about microdosing? What we know to date is that it can have effects, small effects. And we also do not only show enhancement, we also show null effects, like we gave people creativity tests. And in the beginning, I said they take it to enhance creativity. We did not show an effect. Could be due to the tasks we used, or it could be due that it just does not affect creativity. The effects were partly in line with higher doses. We showed that, or others also, that one dose works in healthy volunteers. Studies have shown that it is safe, but we need repeated dosing trials. And a remark I have to make is we showed statistically significant effects, but are they really clinically relevant? And this is what we have to show in future research. So what should be explored? I showed you in the beginning the range of the indications for which people use microdoses. Now we have to find out whether it is true. So we have to move on to patient studies. What I also think is we see effects on mood and attention in healthy volunteers. Then we can have a look at patients who have problems with this, like depressed patients or patients with ADHD. Do they benefit from microdoses of psychedelics? We have a large range of classical psychedelics. We should investigate which psychedelic is suited for which indication. You have short-acting psychedelics, you have longer-acting psychedelics. When do you need what? And does it really matter? I think it can, because LSD has also effects on the dopaminergic system, which psilocybin does not have. If you think about ADHD, you also know that there is involvement of dopamine, so LSD could work in ADHD perhaps better than psilocybin, but we do not know now. We should find out about which schedule is the best to use. Should you use it every day, every 2 days? And we also should look into the safety of repeated dosing, because, as I said, and Gregor too, psychedelics enhance neuroplasticity. But what if you trigger this mechanism over and over again? Will it be good? There are studies showing that, here, the one below, that if you give it repeatedly, you still see this increased neuroplasticity. Another study shows that rodents became more aggressive, looked scruffy after repeated doses. So this is something we really have to look into. And also a last question is, do these people need psychological support? Because they will not have this mystical experience or this mind-altering experience, but still anecdotes show that anxiety can increase by using a microdose, but that can be because latent emotions come to the surface and people might need psychological support with that. Lastly, is it needed, psychedelics? That is a question we have to ask. And I think we need it in psychiatry. Just look at conventional treatments and what they can do and cannot do. We know about the challenges, that there can be poor treatment outcomes or low patient satisfaction. And we now have to find out whether psychedelic medicine has benefits. I'm coming near the end because I've used my time. What is the future? I think we are going to do a trial in ADHD patients, but I know that there are more studies underway also looking into depression. Here's a quick view of what we're doing. And here are my acknowledgements. And I thank you for your attention. And I give the floor to the moderator again.
Thank you so much. It's so interesting to see this area of microdosing moving from anecdotal and survey research really into empirical research, and you're really leading the way on that. And thank you for kind of mapping out where we need to go next in terms of some of the really interesting research questions that are still unanswered. Before we move on to our final speaker of this panelist, I just want to ask the audience to please enter any questions that you have for any of our panelists. We have quite a bit of time at the end of the next talk to answer your questions, so we're really keen to hear what you're curious about. So our final panelist for this first panel is Dr. Guy Goodwin. He is the Chief Medical Officer of Compass Pathways, and is an emeritus professor of psychiatry and the NIHR emeritus senior investigator at the University of Oxford in the UK. His research interests are in the treatment of mood disorders and the potential to improve treatment using new technology and new drugs, notably psychedelics. Please go ahead, Dr. Goodwin.
Thank you very much. If I can just share my screen, I should be possible. There we go. Okay. So far, so good. Right, okay. So the question I'm going to try and answer is whether we can successfully remedicalize psychedelics, and I'll touch a little bit on why I think that's important. I have to start with a disclaimer because I'm going to be making forward-looking statements, as they say in the trade, and therefore I should caution you that the company's position is officially represented in papers lodged with the SEC. My objectives today are to introduce psilocybin a little more than has already been done, but much has been done already, as you know, to just briefly touch on the evidence from healthy volunteer studies, again alluded to by Sid and others, but really quite important for setting the scene and choosing various aspects of the dosing. Look at the clinical trial stories so far, and then finally to look at treatment-resistant depression as a test case for the regulatory approval of a psychedelic drug. So psilocybin is, as you've heard, psychoactive. You also have heard that it's an active ingredient of mushrooms. It has an established knowledge of subjective effects, so there's quite a lot of folk tradition to draw on in how we have started to study it. And generally, it's a well-tolerated compound. Now, Professor Hasler gave us an excellent introduction to the neurobiology. I'm not wishing to restate it, but psilocybin in our hands is a patent-protected formulation. The original synthesis by Hoffman was not actually sufficiently stable or pure to meet modern standards, so that actually Compass Pathways were able to patent the preparation of COMP360, as we like to call it, and that's the drug that what version of the drug that I'm going to be talking about. It's in all respects similar to the psilocybin that occurs in mushrooms, of course. It binds to 5-HT2A receptors, and we believe that's the primary site of action. The relevant neurons on which we think it acts are obviously in the cortex, and the cortex itself, the organization in the cortex is of tremendous interest. And one of the findings, which is quite an inspiration to where to go next in terms of indication, is that the default mode network, which is the pattern of activity you see in the brain during introspection, is quite markedly changed by the administration of COMP360 and other drugs like it. So if you look at this image, it kind of looks like it might have been produced by a creative artist, but actually this is perilously close to science, as close as brain imaging gets anyway. On the left, you can see in healthy volunteers the average effects and brain connectivity under placebo. And the external spots on this circle represent brain areas that are tightly linked to each other and form these networks, one of which is the major one, is the default mode network. And you can see that normally they're relatively sparsely connected with each other. By contrast, on the right is what you see during psilocybin. And during psilocybin, you see that there is a much denser connection between networks that are normally fairly isolated. So that the very experience that we hear about from patients and indeed from anyone who takes these drugs is this greatly increased sense of connectedness. And here you can see in the brain a literally greater increased connectedness between different brain areas. And that we think is maybe very important to all of the effects that we have seen and will see using these compounds. Now, going back to these healthy volunteer studies, the first of these was actually conducted by the Hopkins group, Roland Griffiths, in 2011. And what you can see on the left here is that there's quite a convincing dose response curve when observed by external observers rather than simply reported by the subjects. So you can see it's a very orderly increase in apparent drug effect, which is obviously a mixture of things which strikes someone observing a subject who's taken these drugs, or indeed in the apparent joy or intense happiness that's observed by someone who's taken these medications. And you can see that the dose range that was explored here from 0 to 30 really covers the full range up to quite intense psychedelic experience. But it looks pretty orderly. It looks like we have a drug that has a dose response, and that may be quite important in developing it, as I'll mention towards the end. On the right, you can see the well-being described by the subjects. And this is pretty interesting because it highlights a potential problem with these medications in that, as you can see, two lines. One is a line, and this is subjective effect plotted against dose. But the line which is marked by triangles, the doses were ascending. In other words, the lowest dose, the 0 dose, was administered first and the highest dose last. And whereas in the circles, you can see that it was in the opposite direction. And the effect of this is to flatten the dose response curve very markedly. Now, clearly, there seems to be a dose response curve between, if you take the average of the two, but it's perhaps not as striking as the overall observed effects. And the other aspect of this is that it's much steeper when it's in the ascending direction, suggesting that actual experience of the drug increases subsequent responses to it. And that's obviously going to have implications for how we are to use it in ordinary practice with repeated administration. What are the adverse effects? Because most of the studies that we've done have really only allowed us to be confident about them. Well, I suppose we have to think carefully about this. Conventional adverse effects would be increases of anxiety, fear, panic, a variety of things that are actually part of the experience that people have under a psychedelic. And of course, they're endorsed very strongly by patients who take it. But these are sort of unavoidable, and I think we're going to have to sort of rechristen them, because anecdotally, patients who experience these things often find them subsequently to have resolved in a way that has lasting benefit. So you've got to be a little cautious how you score an adverse effect when using these drugs. In fact, it's very reassuring that on subsequent days, as you can see on the horizontal lines on this graph, you have very much fewer in the way of adverse effects. So that in fact, only headache on the immediately day after dosing appears to be reasonably consistent and predictable. And for the most part, these appear to be safe drugs to take in terms of the medium-term follow-up. Now, what have been the studies that have been done in patients? Well, this shows you the spread, which has gone from 2011, which was really the start of this renaissance, as Sidney indicated, through to the present time. And the UCLA study was actually in patients with cancer. This was kind of the first of these studies. It was very small. It used not very clear entry criteria, other than the fact that people were in extremis as far as their cancer went. It moved on with the two placebo or semi-placebo control studies conducted at New York and in Hopkins and published simultaneously in 2016, again in cancer patients and again addressing existential distress. What was important about these studies was that they also, particularly in the case of the Hopkins study, employed conventional rating scales for depression and anxiety, which allow us to really read across to the potential in other mood disorders. In fact, existential distress isn't really a diagnosis. It doesn't really enable anyone to go to the regulatory bodies and ask for an indication. And that seems not to have been fully appreciated by the investigators when they did these studies. But in other respects, these are good quality studies, and they seem to show quite a marked effect on depressive symptoms. A big reduction at week 1, sustained over several weeks in patients who had considerable and significant levels of both mood and anxiety. The issues of mood disorders more broadly was taken up by the Imperial College team, and Robin Carhart-Harris and David Nutt did an open feasibility study published in 2016 and with a follow-up in 2018 that showed a reasonable response in patients with long-standing and severe depression in an open label study, which is subject to caveats, obviously, that both the investigators and the subjects knew what was going on. And that's been followed by a Hopkins study on MDD, major depression, which used a waiting list control and again showed very substantial benefits in MDD patients. So this forms the background to the study that we felt needs to be done. But in addition, there's been a comparator study recently published where the Imperial College team took the opportunity in essentially an experimental medicine study to compare outcomes for patients receiving 2 doses of psilocybin at 25 milligrams, that's the psychedelic dose, with patients who were randomized between that and escitalopram, which I guess is one of the more effective standard antidepressants, pardon me. And what these graphs show is that, and we think really, in fairness, one should only take the data up to the second psilocybin dose to understand the impact of the drug. You can see that there's a quick fall in symptoms at week 1, very marked delta, as you might say, in the MADRS, and that this effect is larger in the patients who received psilocybin. And the difference was was actually statistically significant on the MADRS, but that actually wasn't the primary outcome. The primary outcome chosen was the QIDS, and on that, this was a difference that not statistically significant. That's not entirely surprising because actually this study was never powered for this particular comparison. So it needs to be treated with a little caution, but the efficacy measures, which are shown on the right, all favor psilocybin, and again suggest that we're dealing with a drug that may be effective in severely ill patients. So where should we begin to try and do a study that is going to be acceptable for regulatory purposes? Now, when we say regulatory purposes, we mean do a study that will be large enough to be statistically significant and convincing, with a standard of performance which allows complete audit and verification of data. None of the studies I've shown you would have met those criteria. It's extremely expensive to do regulatory studies. And for that reason, it's essential that they're done by someone with a lot of money and has raised a lot of money in order to do it. So that is where the story starts for Compass Pathways and for George Goldsmith and Katya Malievskaya, who were the founders of the company 4 or 5 years ago. And they've managed to raise the funding to do the necessary study, and the choice of target has been treatment-resistant depression. This slide very briefly summarizes why it's important. There's a lot of patients who have treatment-resistant depression. It's an area where if you look at all the guidelines across the world, there's uncertainty about what the next step should be. And if you look at STAR-D, which is one of the larger systematic investigations of sequential treatments in depression, the success rate after the failure on 2 medications drops to something under 20%. So there's very considerable need for treatment improvement. And there seemed to the Compass colleagues that psilocybin might be the place where one could start to look at meeting that unmet need. Now, psilocybin is not just a drug, it's a therapy in our hands. As you've heard, it's essential that patients be prepared, and that means establishing a therapeutic alliance, and it requires that the patients understand what may happen to them. During the session, there needs to be someone present, and in fact, there may have to be two people present for purely practical reasons. When in a psilocybin session, patients are really vulnerable, and therefore it may be for safety reasons both ways that you need two people present, perhaps when you think about it for obvious reasons. And then after that session, there needs to be integration. There needs to be some way for patients to make sense of things. We don't know how important that is, and the approach that we've taken at Kuypers is to minimize this wraparound psychological support. So we're not doing psychotherapy, we're doing support, and the patient's experience is largely determined by what happens in the session and what sense they make of it. It's not driven by some hypothesis that the therapists have about what is good for the patient. The place where it happens is important, and as you've already seen, that one has to provide something that is comfortable. Gregor, I think, illustrated a place with a large mushroom symbol. We tend to think of our medication not necessarily requiring mushrooms, but it certainly requires the other elements of things, which is music, comfort, and support. And that's illustrated in this diagram below. Now, the trial that Kuypers designed, and this was some years ago, several of us formed a small group who thought about this over the course of a day and came up with this study. Has recently finally reached its target recruitment. In fact, it overran. It was sufficiently successful to do that, and it went on during the COVID era. So it's a remarkable achievement, I think. This data will be read out within a few weeks, certainly before the end of the year, but I'm sorry I can't give you the results now because I don't know what they are. The preparation, as you can see, was patients were required to discontinue any drugs that they were taking, which is a problem, but in fact was achieved with the right motivation by a lot of patients. And the concerns about SSRI withdrawal were really contradicted by the experience of our patient group, the vast majority of whom found that relatively straightforward with the right support and preparation. At the point that they have withdrawn from the medication, there's a short gap, and they are then randomized to receive either 1 milligram of psilocybin, 10 milligrams of psilocybin, or 25 milligrams of psilocybin. And that's administered on day 0. And there's a reconsolidation on day 1, and then patients are followed out to week 3, which is the primary endpoint for the MADRS, and then a further measure is made, secondary endpoint at week 12, which is essentially there to measure this consistency and the sustained nature of the effect. A comment on the 1 milligram is that this has actually been approved by the FDA as the best way to do this. We felt it was quite important to be able to prepare patients for the fact that they were going to get some dose of psilocybin, even though we knew that 1 milligram would be rather small. You can see that our expectations about the response to that arm of the study are a little uncertain because there is no placebo treatment here as normally understood. Remember that placebo is usually a pill taken every day during the course of a conventional drug trial. This is not happening in this study. They have a single day when they have a rather minimal experience compared with what they expect. So there may well be quite an appreciable nocebo effect in this design. It's a new territory, it's a new area. We're going to have to work out how the best way is to do these studies. But this, in the first instance, is what we've chosen to do. How are we going to take this forward? Well, if we have a home run result, we shall look very carefully at how we go forward, but there are a number of other studies that are taking place, one of which is to look at the effects in patients as an augmenting treatment in patients who are continue to take an SSRI. And we also have succeeded in randomizing about half of our acutely treated patients into a follow-up study, which will give us some idea of the durability of the effects in patients who've done well. What happens after that is very contingent upon the results of the study that we'll read out later this year. If they're successful, then we will take forward potentially other indications, and this gives you some idea of the preliminary work being done predominantly by investigators who have been supported in part by Compass Pathways. And those indications are, as you can see here, chronic cluster headache, bipolar disorder, body dysmorphic disorder, anorexia nervosa, and some aspects of depression like suicidal ideation, which are also of interest. So I'll stop there and thank you very much for your attention, and end up with a quote which is from a patient who I've met from the Imperial trial. And just to say that one of the striking things, one of the things that give us conviction in this area is the testimony of individual patients. And it's key to remember that what we're doing is trying to restore access to these substances that were so sadly lost those years ago, as Sidney described. And we believe the only way for that to happen is to get the sort of regulatory approval which will allow everybody, including poor people in healthcare systems that allow reimbursement, to get access to this treatment. Thanks very much for your attention.
Thank you so much, Dr. Goodwin, and thank you to all of our panelists for a wide variety, from the history to the biology to where this research is going, and a nice reminder on that last slide of really why we're all doing this. I'm going to begin with a question. I'm asking the audience to please enter any questions that you have, and we'll get to as many of them as we can. I'd like to begin with a question that's directed at Kim, so it's more related to microdosing, but any of our panelists are welcome to answer this question. And it's just to talk a little bit more about the placebo effect in the context of using psychedelics and microdosing. So how do you address the placebo effect, and how can you differentiate between sort of the active substance effect and placebo effects?
That is the question, and I forgot to address it in my presentation, because there is a study that has been done by Imperial College, and they did a self-blinding study, naturalistic study, and they showed when people took microdoses and when they took a placebo, both groups improved. So it was not due to the microdosing, they said it was due to just improving over time, and also partly they said it is a placebo effect. But we also know about other studies that have shown that people can expect certain things by reading it, but then show different effects on behavioral tasks. So I believe that that is bad for a researcher to say that. I believe it's not only a placebo effect. And I think the best proof is that we show on a biological level that there are effects that you do not see when you take placebo. So enhanced neuroplasticity, you also see it in the studies that have been done by the Chicago lab, that there is a change in neural activity. It is a difficult topic, but I do think that the data speak for themselves.
Great, thank you. I think this brings us to sort of a related question, which the field is really grappling with, which is the problem of blinding in psychedelic trials. So I'm wondering if any of you kind of want to comment on how do we approach studying these when it is much harder to blind. This is more for macrodoses. So when you're giving a full dose of, say, psilocybin or MDMA, it's hard to find an option for your control group that people don't know that they're getting the placebo. So I'm curious what our panelists think about that and how we kind of grapple with that. Any thoughts?
I'd like to say something. I mean, one way is giving a lower dose. So you tell the patient can be any dose, and they give a high dose and a low dose. The other thing is you give a substance with some psychotropic effects like Ritalin or so. Another way, I think that these are two options, but certainly not a perfect option for these acute effects because they're so strong. But these would be two options.
Mhm.
I agree with with both options, and but I also have my doubts for the second option. I think it's beautiful that you can give a range of substances, but you also have to prepare a subject for a psychedelic, not so much for receiving Ritalin or something else. So, but potentially these researchers will include this then, huh?
Yeah.
Question mark.
Yeah. I think we're seeing like the low dose 1 milligram comparator become more commonly used, but we're seeing, as you showed in your research, that actually has a like that microdosing can actually have a neuroplastic effect. So it's probably also doing something. It's not a perfect
Yes,
comparator
yeah.
for true placebo.
So for 1 milligram of psilocybin, we don't know, but I also read in these papers that they say it's an inert comparison or comparator, but I'm not so sure about that, because we show with 5 micrograms of LSD that it also has effects on neuroplasticity. And another interesting thing, I think, is, but that's more something for microdosing research. There are groups that are using niacin as a comparator. And we also know that niacin, if you take it repeatedly, it has positive effects. So I would also not advise that using it in microdosing studies. In macrodosing studies, I don't think it's that problem if you just receive 1 dose of niacin and vitamin B.
Yeah. Sid, did you want to add something?
Well, I think this is actually a question that really applies throughout much of psychopharmacology in general. And there's an interesting discussion following Mitchell's recent publication with MDMA, where the question of the placebo effect is raised. And actually 2 University of Toronto psychiatrists, Burke and Bloomberger, have part of the dialogue back and forth on this, where they argue that one position to take is that, yes, there is a placebo effect, and there's probably a compounding active effect. And if in fact the treatment works, why don't we just get on with it and not get too caught up in trying to separate out what is the placebo effect and what is the active drug effect?
Yeah,
Yes,
could I support that view? I mean, if we're dealing with conditions that have very low response rates to the conventional treatment, and we're able to substantially exceed those response and remission rates, then in a sense we shouldn't get too hung up on what the placebo condition means. I mean, it's the meaning of the active condition that really matters. But a lot of it hinges on what the results are going to be of the studies that meet the necessary standards, actually.
Yeah, I agree. You know, the signal, our effect sizes are large, the signal is strong, like we need to proceed. I have a question here. If the experience with psilocybin is really one of the top 5 most significant experiences in the life of somebody, doesn't it imply an extremely high addictive potential? Don't patients want more of that? Can our panelists address that one? Who would like to tackle that first?
That is the question. Psychedelics are used to combat substance disorders, so we know that it is a therapeutic agent in these conditions. But I also get what you mean. I think they are not physiologically addictive, because they also they induce immediate tolerance. But I do think that some people might have some kind of psychological not need, but perhaps wanting to take it again, to experience it again. But I do not know whether that is, I don't think that it's the majority. If you talk to people, they most of them take it really thinking it through and only take it once a year, or leave more in between. But there are people, if I look at our naturalistic studies with ayahuasca users, there are people that have used it 300 times in their lives. And then I'm also asking why. But I do not think that there is such a risk in it. But I also would like to hear Gregor and Guy on that matter.
Yeah, what are you guys noticing in your work?
And I like to compare it to like going on Mount Everest. You know, that's an amazing experience, unique, but when you go down, you don't feel like going up again the next day. So I think it's very similar. And I think these people using 300 times ayahuasca, usually if I talk to them, they don't have a strong experience. They have a very weak experience. They're looking for the strong experience. But you really have this Mount Everest experience with all the really crazy and you're exhausted, everything. It's an experience for your life, but you don't want to do it again. Also with children, I mean, many people get addicted to having children, but mostly if you have 2 or 3 children, then yeah, that's a life-making experience, but you don't want to have it like 100 times.
I think we're going to have to be very cautious about this, actually. I think many of us had the experience using ketamine clinically, that we get to a position where patients only really respond to ketamine, and we find ourselves giving it weekly. And it was really not developed to be given like that, and it's an uncomfortable feeling. And I can quite foresee that there will be potential problems in using psilocybin or related compounds in patients who have very refractory conditions, and for whom really this is the only treatment. And we're going to have to wrestle with, you know, how frequently it's legitimate to give this. But I think that's a bit different from what really is implying a sort of recreational thing about how often people would want to take it in a completely free access. And I think one of the advantages of keeping it a regulated product will be that at least some control can be exercised over that.
It will also be about giving them information and creating expectancies, also telling them that this is something like a social or therapeutic lubricant that might set off a therapeutic process, but that you need this traditional therapy, like cognitive behavioral therapy or psychotherapy, to work on their condition. Perhaps this creating of a mindset will also help them not wanting the drug too often.
I think at the moment, anecdotally, certainly in this country, that there's more of a problem with people munching mushrooms on a regular basis than actually access to, you know, as yet medicines. And you know, I think the difficulty with microdosing is I can see that that's quite a slippery slope to using them very frequently, rather than actually the full doses, which as have been indicated, don't really lend themselves to repetition, because you get this quite marked habituation or desensitization of the system to the very big dose.
Yes,
so,
but yeah.
I think that's an issue. I mean, for the people who want to legalize it, and I think that's a, you know, particularly an observable phenomenon, actually.
But for when you talk about microdosing, I also think it might have its benefits and not being addictive. We don't know how long the effects of a full hallucinogenic dose last. We don't know in a therapeutic context. Huh? So it might be that future therapy will look like giving a full dose, working on the person, and then going to microdoses because you just need this neuroplastic effect. We don't know.
It's possible. I mean, what I'm saying is that what is actually happening is that people are being told it's completely safe to take mushrooms. They're taking it. We don't really know it's that safe, particularly at higher doses, because there is actions of the 5-HT2B receptor, which are associated with cardiac abnormalities. And, you know, that means that you really have to be quite cautious. And the reason we have regulation is to make sure things are safe and effective. And that's why it probably needs to go through that pathway to be safe and effective.
I fully agree. Yeah.
In other aspect, it's certainly the selection of the patient. I mean, we screen them very closely. If they have any problem with addiction, then we exclude them from our studies or treatments.
I think, Dr. Kennedy, go ahead.
Well, I just have a comment and a quick question. I think it's important to differentiate between use from time to time without dose escalation, which I think most people would say we don't see much of, as opposed to tolerance. But the other question, and Guy, I think you touched on it, this question of whether you need to withdraw SSRI or other standard antidepressant treatments when you apply, right now for clinical trials, I appreciate that we do not want combination treatment. But do we actually know the safety, the potential negative effect of the combination, or in fact, might there be a synergy?
Well, I mean, we've just completed a study, a small study of 20 patients on SSRIs who have had the full dose of psilocybin, and we'll tell you pretty soon what the results are from that.
Okay, so we got a lot more questions coming in here. I have a couple questions for Dr. Hasler, but again, anyone's welcome. There's a question on the role of psychedelics in inflammation, and curious if there's also any interaction between the anti-inflammatory effects and neuroplasticity.
Yes, there are some, I think, smaller studies showing that psychedelics have anti-inflammatory effects. And sure, they may be related to the neuroplastic effects, but I'm not aware of studies really showing this relationship. But I think it could be potentially, true, that this inflammatory aspect, which is not always prominent in depression, that this is really a factor that leads to these problems or reductions in neuroplasticity. But I'm not absolutely sure about this relationship.
It's actually a very interesting question, because the inflammatory biomarkers may actually be quite useful ways of getting at drug action, and supportive to get around this placebo problem what we were alluding to. If you could show robust effects on on immune biomarkers that are associated with depression, that would help to support the conclusions that we reach from measuring symptoms. So I'm very bullish on that. I think it's quite important to think about that.
Next question. Are there any psychiatric illnesses where psychedelics might cause more harm than good, and where we might need to use caution? And how might we develop standard guidelines around that?
What's the experience in Switzerland, Gregor? I mean, Switzerland allows compassionate use, doesn't it?
Yeah, they do. So we exclude a patient who had any kind of psychotic episodes in their history, and also patient with a family history of psychotic disorders. So because there it may cause harm that the patient is already very magically thinking, or problem with structuring thoughts. This may kind of cause confusion, for example. Also, I think people with really low education, low IQ, sometimes I think they feel confused or they have more problems to integrate it. I think that's going to be a really important question, if this is really just for some people with some education or for everyone or a cultural background. But certainly we just exclude psychosis and family history of psychosis. So I have experiences bipolar disorder that can be helpful, also with autism or autistic personality. So it really has a wide range of indications in my view.
Sid?
Yeah, actually, I saw Professor Brian Rush was there. If he's still there, I was thinking that he has extensive experience and recently reviewed a lot of this area, so he might want to comment on the who not to treat. Any thoughts, Brian?
Yeah, thanks, Sid. I think there's not a lot of consistency other than around psychosis. And we did turn up, I think, 1 or 2 studies looking at psychedelics with schizophrenia. But in general, I think that's the guideline. And then I also heard the question around the SSRIs. The experience in Brazil with the Santa Daime Church which have like 10, 15, 20-year histories of use of ayahuasca, like people with that length of history, they do not exclude people, and they report very, very few, if any, like it's just like not part of their exclusion criteria. I think that's interesting looking at long-term use, but in general, that's also an exclusion criteria. So I would agree, we probably don't know enough, but that history out of Brazil and these long-term users of ayahuasca in particular, I think it's noteworthy that their experience is not really required to as an exclusion criteria.
I think
I was thinking
there's an internet survey suggesting that lithium also is probably a counterindication, that you may get, there's an association with seizures, I think, in potentially with and there is in animals you get a sort of serotonin syndrome type thing with lithium added to 5-HT2 agonists. So I guess that adds to the list, unfortunately, since I'm interested in bipolar disorder.
Kim?
Yeah, it's when Gregor was talking, it reminded me of something, actually a very important question that we have to answer, I think, in the future, is whether the mystical experience is that important. We have these novel psychoactive, non-hallucinogenic agents or analogs of psychedelics that do not cause these hallucinogenic effects. So they might be suited to give to people who cannot understand such an experience. But then we have to understand the biological mechanism and understand whether that is causing improvements of symptoms.
Good question. Yeah.
We have a question that's sort of related to this conversation, is about sort of the use. Typically, people are weaned off of their SSRIs in the psilocybin research. Is there any evidence of what happens if people stay on them? Is that dangerous in some way, or does it reduce efficacy?
Well, the belief is it reduces efficacy. But I think, as Brian was just indicating, that's may well be wrong. That's why we actually treated these 20 patients of ours who were taking SSRIs with psilocybin, just to try and find out.
Okay. Next question is around, is this a one-time thing or something that people are going to need to have repeated sessions of? I think we've seen in the media at times that this is sort of, especially for the treatment of depression, almost not marketed, but like the media is saying this one big brain reset. But actually, I think, in real life, that may not be the case. So I'm curious what you guys think. Is this something that people are going to need over time?
Yeah, I mean, in Switzerland, we have this long-term experience because over many years we have this compassionate use, and there are many patients, they only have once this, and this enhances the psychotherapeutic process, but many do repeatedly. So usually have a 3-month interval, and then we do it again. And then some people, let's say, do 2 years, 8 times, or many patients like 4 times. And it's really important that's always embedded in a psychotherapeutic process. That's a bit of my experience.
Yeah, we don't have a lot of the long-term follow-up data yet. So that'll be really interesting to see what's happening to some of these patients after their enrollment in the psilocybin trials.
Yeah, for MDMA, for example, not under the umbrella of classical psychedelics, but still in the spectrum, we know that people who were in these trials, some of them would like to have additional sessions, so to work more on, they see the benefits, but then still they want to work more on their trauma.
Yeah, and sometimes,
so I think, yeah.
Sorry, go ahead.
No.
Yeah, I was going to say, sometimes it's just that the structure of the research doesn't actually match what that patient needs. They may have needed more than like the 3 MDMA sessions that we're able to give them in order to get a greater resolution.
I would just make one quick comment, and that is that we're talking as if we're talking about homogeneous disorders, whether it's depression or any of the other disorders. And so, for example, if you look at that lovely Carhart-Harris cartoon figure, where we see the profile of SSRIs causing some benefits, but including emotional blunting, which is no stranger to Professor Goodwin, but also the idea of the openness of the psychedelic 5-HT1 activity, 5-HT2, There are presumably individuals where this opening up of the default mode network is actually a very significant benefit, but perhaps there are others where that's not such a relevant mechanism. What comes to mind included is the concept of depression with strong anhedonia versus those who don't have the same level of anhedonia. So there may be different populations who will do very well and stay well, and there will be others who need sort of frequent top-ups, if you like.
Yeah, and I think that brings us to sort of questions like towards personalized medicine, which is hopefully we'll be able to go in that direction. I think increasingly we are. So on that note, we're going to end our first panel. Thank you to all of our panelists and to the audience for asking really interesting questions. So we're going to take a break now, and we will return at 2:20 p.m. Eastern, which is 11:20 Pacific and 8:20 in CET time. So look forward to seeing you guys back in 10 minutes. Take care.